Good morning, class. We would like to start a lecture on basic biochemistry, and yes, so our course outline. At the end of the lesson, students should be able to describe the chemistry and properties of lipids. Also, lipid chemistry, that is classification of lipids, saponification of lipids, iodine number and its importance. But the iodine number, I may touch on it or not. Biomolecules of living cells. A biomolecule, as previously explained or defined, is a biological molecule used to describe molecules present in organisms that are essential to one or more typical biological process. Examples are proteins, carbohydrates, lipids, nucleic acids. Lipids are biomolecules that are hydrophobic in nature because of the high amount of hydrocarbons in their structure. So when we say that something is hydrophobic, it means that it is afraid of water. So look at hearts of oak. They call them phobia because they are afraid of Asante Kotoko. So let's see, hydrophobic. Why are they hydrophobic? Because they are afraid of what? Water. So let's see, CH3, CH2, CH3. This is called propane. Propane is what? Afraid of what? Water. So it is called, it is what? Phobia. Phobia. It loves water. It loves water, then its name will now be called philic. That is filial love, brotherly love. Philic. So hydro means water. So hydrophilic. Also called filial love. There is brotherly love. Brotherly love is also called filial love. Like the love between Hannah and Sigdo. So let's continue. So, like I said, lipids are biomolecules that are hydrophobic in nature because of the high amount of hydrocarbons in their structure. They are relatively insoluble in water, but readily soluble in nonpolar solvents such as chloroform, benzene, and ether. So when we come here, you realize that this propane will be insoluble in water, but very soluble in what? Alcohol. Ether, because they are all hydrocarbons. Benzene, because they are all hydrocarbons. Let's proceed. So this is the lipid bilayer fluid mosaic level, fluid mosaic structure. So let's see. This is a lipid bilayer, and with the lipid bilayer, we have what? Water surrounding the cell. Water surrounding the cell, then water inside the cell. So we assume that the cytoplasm inside is water, then outside the cell, which is where the interstitial tissues are, is also water. So this is hydrophilic or polar head, and this is what? Hydrophobic or polar tails, non-polar tails. So these are fatty acids. These are fatty acids. These two, these things. So you can see that this is what? So when we look at the lipid bilayer, we have two polar heads making a membrane. Polar heads making one membrane that is at the outside of the cell. That's the plasma membrane. Then inside the cell to another polar layer to interact with the cytoplasm or the cytosol, the fluid in the cytoplasm. Then in the cell membrane itself, or plasma membrane, is made up of what? Fatty acid conjugated with what? Glycerol, making diacyl glycerols. So you can see the structure here. We'll go further deep into it. So non-polar tails are hydrophobic. They don't like water. They're afraid of water. So if you want to send something inside the cell, water cannot go inside the cell unless it is facilitated or unless something helps it, and that is what we call active transport. And it has to be helped by something to overcome the concentration gradient and to overcome the fat over here, the lipid over here. Okay. So examples of lipids are fats and oils, steroids and waxes, fat-soluble vitamins, and vitamins A, D. Fat-soluble vitamins are called ADEK or ADEK. But water-soluble vitamins are the BC, vitamin BC. We have the BC vitamin, vitamin B. That is your energy giving food, gotten from your energy giving food. Vitamin C, ascorbic acid. Vitamin C, which is ascorbic acid. Big acid. So vitamin C is called ascorbic acid, and vitamin B is the energy giving vitamin. And that we have types of vitamin B. We have the thiamine hydrochloride. We have a folate, the one we get the folic acid from, cyanocobalamin. All those things, it seems I taught it in my enzymology when I was teaching cofactors. So let's proceed. So let's proceed. So when we say fatty acids are aliphatic carboxylic acids containing long hydrocarbon chains that may be saturated or unsaturated. Fatty acid has both hydrophobic and hydrophilic properties. Thus, they are amphipathic in nature. So let's see. Let's draw a fatty acid. So let's see. We have this one. Let's clean this. So let's assume that we have fatty acid. Okay, so we have 3 C H C H so we have the C double bond O, then we have the C O H. This is called propanoic acid. Propanoic acid. So we can see that we have the propanoic acid, we have the this is CH3, we have 1 carbon, 2 carbon, carbon 1, carbon 2, carbon 3, carbon 4. Okay, so this is the functional group. The functional group is what? This one. This is what is making it a carboxylic acid or fatty acid. It is also written as COOH. It's the same. This is the same as what I've circled here. So you can see that carbon. This is this is butanoic acid 4. You know, if it's propanoic, then the carbon atoms are 3. This is butanoic acid, butanol. Butanoic acid. The acid is already here. The butanoic acid. So this group is a polar group. Polar means that it can react with water. It can mix with water or react with water. But this group is non-polar. That the CH3, CH2 is non-polar, meaning that it is hydrophobic, afraid of water. Here is it, CH3. No, this one, now we can put CH3 here, it's CH2 because it's being occupied by so CH2 before this group will come and join. So this whole thing is what? It's called an alkyl group. It's called an alkyl group. So this is what we have. So this whole thing is the alkyl group, which is what? Hydrophobic, afraid of water. And this is hydrophilic, because it loves water, so it can react with water. That's why you cannot mix water and fats, but you can mix kerosene and petrol because they resemble each other. Let's proceed. So when we talk about unsaturated, saturated is what? Having double bond or triple bond. But unsaturated is having double bond or triple bond. Saturated is having what? Only a single bond. So saturated. When you say a fatty acid is saturated, it has only a single bond. It doesn't have any double bond. Then this one is unsaturated. The sat is SAT. This is unsaturated. Unsaturated. Let's proceed. So fatty acids are both hydrophobic and hydrophilic properties. Thus, they are amphipathic. So we have the hydrophobic here, hydrophilic here. So they are called amphipathic, having both hydrophobic and hydrophilic groups. So we classify fatty acids as saturated. Saturated means that it does not have any double bond. Monounsaturated, it has only one bond, and this is it. Polyunsaturated, it has two bonds, or more than one bond. So here 2 bonds, it can be 3 bonds, 4 bonds, 5 bonds going. So this is a saturated fatty acid. We can see that there is no double bond. Unsaturated fatty acid, we can see that it has only one bond. Forget about this bond because this is the functional group. That's the alkanoic acid or carboxylic acid group. This one is not the one. This is labeled in red because that's the functional group. But we are looking at the saturation or unsaturation in the side chain or in the alkyl group. That's the hydrocarbon group. So further examples, we have butyric acid contains 4 carbons. It has no saturation, so it is a saturated. Has no unsaturation, so it is a saturated fatty acid. We have oleic acid, has 18 carbons, and it has a carbon double bond here, so it is a mono unsaturated fatty acid. 2 double bonds are here, so this is also 18 carbon compound, but because it has 2 double bonds, we call it linoleic. Linoleic acid is a polyunsaturated fatty acid. Let's see cis and trans fatty acids. So you can see where the 2 carbons containing the double bonds, the hydrogens that are attached to them, are they on the same side? If they are on the same side, it is called cis fatty acid. If they are on the same side, the whole compound is a cis fatty acid. If they are on opposite sides, this one is pointing up and the hydrogen here is pointing down, it is trans fatty acid. So let's see, cis fatty acids have bent shape, whereas trans fatty acids are more linear. So look at the cis fatty acids. The hydrogens are the same side. So it is bent. Gravity is bending it. Then trans fatty acids are linear. There is no bending. You can see where it has been highlighted. There is no bending. So the ladies who like fried rice, you can see over here that fried rice is really bad. Some of them, they have graduated from fried rice to what we call Kentucky Fried Chicken or KFC, and they will be worrying their boyfriends and their husband. They will be worrying their boyfriend because they cannot worry their husbands. By their boyfriend, they will be worried. So these ones have trans fat, which is bad fat, and these ones have saturated fats, not as good fat. But if you have fruits, olive oil, tilapia, almond, cashew, they will give you monounsaturated fats, which are very good because they contain omega-3. So let's look at what these fats do in the body. So if you are eating fried rice, Kentucky Fried Chicken, and those things, look at this. This yellow coloration is what? Fat deposits in your blood vessel. So this is your blood vessel supplying the heart. So if you have a vein, this is your blood vessel supplying the heart. Or so this is atherosclerosis, where blood cells are inside and fat has accumulated. So it will be blocking the free flow of blood to your blood vessels. So we call it atherosclerosis. Nomenclature of fatty acids. How do we name fatty acids? So we have the delta method and the omega method. But let's start with the delta method. So with the delta method, these are 18 carbon fatty acids. 18 carbon. Please, let me explain something briefly. We have yes, we have this. If you have this is a fatty acid. This is the same as CH3. Okay, let me label like this. Carbon 1. So this is carbon 1. Carbon 2. This is also a carbon. This is a carbon. This is a carbon. This is a carbon. So any joint you see is a carbon. And it is H is here. H is here. That's hydrogen. H is here because the stability has to be maintained. H is also here. H is here. H is here. I hope you are appreciating the structure. H is here. H is here. H is here. H is here. Okay, so, and this is what CH3. 1 H here, 1 H here, 1 H here. So this is the same structure. Anytime you see a joint, it's a carbon. So this carbon is inside this joint. This also here. Please, do you understand this? I know you understand. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, carbon 7. So this is 7 carbon. So it is called heptanoic. Heptanoic acid. It's a fatty acid. Heptanoic. So it contains 7 carbon atoms. Let's proceed. So let's name this carbon, this structure. Let's name it. So 18 carbons. There's a double bond here between carbon 9, that's carbon 9, and carbon 10. So this represents the number of carbons. So we have 18 here. Number, whenever you see this hash sign, is number. So number of double bonds is 1. The delta means the carbon atom containing the first carbon atom containing the double bonds. So this is the first carbon atom. We started naming the carbons from the functional group. That's the carbonyl carbon here. So 1, 2, 3, 4, 5, 6, 7. So carbon number 9 is what? Here is the delta 9. So let's name the others. So here we can see that we have 18 carbons. 18. So we have 18. Number of double bonds is 1. Then the delta 9, meaning that 9 is here. That's the first carbon of the 2 carbons bearing the double bond. It's 9. It's 9. So we have 18, 1, delta 9. Then we bring the name 6, 9, octadecanoic acid. We also have, this is a what? Eicosapentanoic acid. It has what? 20 carbon atoms. But here we are going to use not the delta but the omega. Omega is W. So we have, for the omega, the numbering starts from what? The numbering number 1 starts from the latter end. Then the highest number of carbon is on what? Is on the functional group. So, but now we haven't gotten to the omega series, so let's still use the delta. So for the delta, we have 20 carbon atoms. So we have 20 carbon atoms. Then the functional groups, unsaturated, the unsaturation, that is the double bonds, are 5. So 5. Then the double bonds, the first carbon bearing each double bond is 5, number 5, number 8, that's the first carbon, number 11, number 14, and number 17. So they are here. So the name is eicosapentaenoic pentanoic acid. Nomenclature of fatty acids. So now we have the omega series. So omega series is the same, but just that instead of numbering from here 1 to the last, we start numbering from the last carbon rather to the functional group. So the functional group bears the highest number. So here the number of carbons is 18. Then the number of unsaturation, this one, the double bond, is 1. Then omega 9, that is, you start counting from the least carbon to the first carbon bearing, the first carbon of the 2 carbons bearing the double bond. So that is 9. So 18, 1, omega 9. Okay, so this one we have arachidonic acid. So 20 carbons. So we have C20, number of unsaturation 1, 2, 3, 4, 5, 5 omega 3. That is, we are not starting the numbering from 1, 2, 3, 4, 5, but we are starting from the highest carbon, which is 1, 2, 3. So the first carbon bearing the double bond is number 3. So we have docosahexaenoic acid. So this bears 22 carbons, 6 double bonds, omega 3. The double bond starts from the first carbon, which is 3. The same follows for arachidonic acid, which has 20 carbons, 4 unsaturated bonds, then omega 6, meaning that the first carbon, the first unsaturated bond or double bond is carried by 2 carbons, and the first one is 6. So 6 is here, we have 7 here, but 6 is the most important because we started numbering from here to 1, 2, 3, 4, 5, 6. Yes, so let's proceed. Examples of fatty acids are here. So for your own relaxation, go and look at the examples of fatty acid. We have, if a fatty acid contains 4 carbons, it is called butyric acid. If it contains 12 carbons, it's called lauric acid. 14 carbon, myristic acid. 16 pyritic. Then if it is a mono unsaturated, if it bears only 1 double bond, we call it oleic acid. If it bears more than 1 bond, that is 2 bonds, 3 bonds, 4 bonds, 5 bonds, 6 bonds, and the number of carbons are 18, 20, 22, then these are the names. So these are, if you take these food ingredients, these fats, oils, and whatever, look at their sources of fatty acids. Look at the percentage of fatty acids they contain. So use this legend or key to study this. Okay, essential fatty acids versus non-essential fatty acids. Essential fatty acids are unsaturated fatty acids, meaning that they have double bonds that are essential to human health but cannot be manufactured in the body. Yes, the body cannot produce them, but the body requires it for its own normal metabolic processes. So, and a non-essential fatty acid is a cheap acid. That is, there are various acids that are required for normal health and growth that can be synthesized or produced within the body or derived in the body from essential fatty acids. So they can be derived. We can produce them in the body, or use them to produce other fatty acids in the body. They cannot be synthesized or produced by the body for the non-essential for the essential fatty acids. That's why they are important, so we have to buy them from the market. But for the non-essential, it is cheap because our body can produce it. We have two types of the essential fatty acids. That's linoleic acid and alpha-linolenic acid. Then the non-essentials include arachidonic acid. This is arachidonic, not arachidic. Stearic acid, then palmitic acid. So arachidonic acid is spelled like this. So go and change it. Arachidonic means 20, arachidonic acid. Let's continue. Effect of carbon length and double bonds. The length of the fatty acid residues and the number of their double bonds affect the melting points of the fats. It's true. A shorter fatty acid with more double bonds will require a lower melting point or a lower melting temperature, meaning that if the fatty acid is shorter and it has many double bonds or plenty breast, it means that small proposal from a guy he will accept. So the short ladies who have plenty breast, when a guy proposes, no, whether you have money, you don't have money, they will accept. So saturated fatty acids, this is the formula. We have common name, melting point. So you can see from this table that the higher the carbon atom, this is 18, 19, 20, that's arachidonic acid, it has a very high melting point or melting temperature. The same for and these are saturated, they don't have double bonds. So the same for stearic acid is 18 carbons, so it requires higher melting temperature. So but this one, CH3 lauric acid, is only 12 carbons, so it requires very low melting temperature. For unsaturated fatty acids too, the more the double bonds and the lower the number of carbons, the lower the temperature it melts. So this one, look at this one, temperature, the carbons are small. Here the carbons, we have CH2 here, CH2 is more than this, so its melting point is higher than these ones. You know, -11 is lower than -5, -49 is lower than -11. Then when you come here to 7, this one has more carbon and lower number of bonds, so it requires high temperature. That's palmitic acid. That's about palmitic acid has 1, 7, that's 8, 9, 10, 15, 16 carbon atoms. That is our palm oil, so it requires 0 degrees to melt. That's why when you leave palm oil at room temperature, it is a liquid. Fatty acids and glycerol, they are called triacyl glycerol. A triglycerol is an ester derived from glycerol and 3 fatty acids. Triglycerides are the main constituents of body fat in humans and other vertebrates, as well as vegetable fat. So they are very important. So triglycerides are our fat. So we have the glycerol backbone. This is CH2, CH2. Then we have COH, COH, COH. Please, this one Okay, so this is correct. Okay, so this is glycerol. Then this one is a fatty acid. Fatty acid. So the R is the alkyl group, and the alkyl group can be CH3, CH2, CH2. But if we put it here, it will be long, very long. That's why we left it as R. And this is the functional group. That's the carboxylic acid group. That's why we have made a bracket around it. So it is bonded to CH2, this one, and we have removed the water, because let's look at the reaction clearly. So we have this CH3, CH2, CH. So let me give you another one. Let me give you another one. So we have, if you have CH3, CH2, CH2, COOH. This is the alkyl group, so we call it R group. That's the R group. And this is the carboxylic acid functional group. Carboxylic or fatty acid group. Carboxylic acid. Group. Yes. So please, this one is going to react with water. Water is H2O. So this OH, sorry. Okay, so water is, this one will react with sorry. So the R group over here, the R group, this carboxylic acid, this is what 1, 2, 3, 4 carbons. So it is butanoic acid. Butanoic acid will react with, butanoic acid is going to react with butanoic acid is going to react with glycerol. Glycerol is the same as alcohol, and alcohol is ROH. So glycerol, so let me go for glycerol. So glycerol is an alcohol with 3 carbons, then 3 OH. So this is glycerol. OH, C, H2, OH, OH, OCH, then another CH2, OH. So we want to remove this OH and we want to react this OH with this functional group. So when we are going to react this OH with this, what will happen is that we are going to have what? OH plus OH will give us water, H2O plus O. So in the end, we are going to have, this one will remain, this C double bond O will be there, nothing will touch it. Then water contains 2 hydrogens, so 1 hydrogen, 1 hydrogen, then 1 oxygen. So we'll borrow 1 oxygen here and add it to water. So it will be left with only 1 oxygen, that is this one. This one. So in the end, we are going to get a structure like this. and it is an ester bond. This one will remain, then this one will also remain. Then this is our alcohol, which is what? The glycerol. Then this is our R group. This whole thing, we call it R. So that is it. So this one, this functional group over here now is what? An ester. This is arachidonic acid, but this one now becomes an ester because it becomes an ester. Ester bond. So it becomes an ester bond because a reaction from it's a reaction between fatty acid and alcohol gives us an ester. So ester. So we have an ester here. So if your name is Esther, you are here. So we now this is the glycerol backbone. This is the glycerol backbone. So we have the glycerol backbone here. We have the glycerol backbone here. This is our glycerol backbone. This is our glycerol backbone. Our glycerol backbone. Then the ester bond. We bring one fatty acid here. This whole structure is a fatty acid. This one is a fatty acid. So we bring it here, over here, to come and bond with this carbon. So we have ester bond. We bring a second one to come and bond with carbon 2. So we have another one. We bring a third one to come and bond with carbon 3. So we have triacyl glycerol. If it is two fatty acids, those in the yellow, they are fatty acids, then it is diacyl glycerol. If it is one, it's called monoacyl glycerol. Then this is glycerol. I know you all know glycerin. Glycerin is what we are talking about here. Glycerin that has been bonded with what? OH and transformed to alcohol is called glycerol. So we can see this is a glycerol backbone. Then the fatty acids are here. I know you remember these structures. Fatty acid. So 3 fatty acid chains. Glycerol. So this is a triacyl glycerol. So this one is a mono unsaturated fatty acid because it has only one double bond. And this is a polyunsaturated fatty acid. It has 3 double bonds. And this is a saturated fatty acid. It has no double bond. Next slide. So this is a monoacyl glycerol. The acyl group is the fatty acid group. So whenever you hear acyl group, that's the fatty acid. The RCOO, R C double bond O CO. And this is the glycerol backbone. Glycerol carbon 1, carbon 2, carbon 3. So it's a monoacyl because it has only one fatty acid bonded to it. Diacyl, it has 2 fatty acids bonded to carbon 1 and carbon 3. So hydrolysis of triacyl glycerols. How do we break down fatty acids? If you have triacyl glycerol, that's a glycerol containing 3 fatty acids. When you treat it with lipase by adding water, when you add water to this compound and add an enzyme which breaks down fats, you end up releasing 1 fatty acid. So it will be left with what? 2 fatty acids. So it's called diacyl glycerol. 2 fatty acids bonded to glycerol backbone. Then if you add another water and add the enzyme lipase, another fatty acid will be released. So it will be left with monoacyl glycerol. Then if you add water again plus the enzyme, the last fatty acid will be released plus the glycerol backbone. So this is how we hydrolyze or break down triglycerides or triglycerides or triacyl glycerides. These are the biological rules of lipids. Lipids or fats are broken down in the mitochondria to release energy in the form of ATP. We call it beta oxidation of fatty acids. Then the fish in the sea is 0 degrees, but the fat will cover the epidermis, the subcutaneous tissue of the fish. In humans, that subcutaneous tissue, which is composed of fat, is called campus fascia. Campus fascia, and it protects the skin, helps in thermal regulation or heat regulation of the skin. So the what we call it, cytoplasm, we have fat also being forming plasma membrane of most living organisms. That's the plasma membrane. So it forms the plasma membrane that protects and regulates the cells in the body. The cells in the body, that's the barrier between the extracellular section of the cell and the intracellular. The intracellular is the cytoplasm. We also have fats also regulating or being coenzymes, apoptotic coenzymes. We also use the rods and cones of the eye. We have the fat also controlling vision. Classification of lipids. There are two ways of classifying lipids. We classify them on their solubility or their size and structure. We can also classify them based on their hydrolyzable activity, or their ability to break down, or their inability to break down. So we have hydrolyzable lipids and non-hydrolyzable lipids. So for the hydrolyzable lipids, we have esters, fats, waxes, steroid. We can break them down. Why can we break them down with water? Because they have a lot of polar functional groups. We can see phosphate is negative charge here. We can see the carboxylic acid is negative charge here. So we have to, but these ones, those which are used to produce the progesterone, the estrogen, the hormones in the body, most of them, they have only the carbon chains and the double bonds, whatever, but they have a limited or small number of polar groups, so they are not easy to break down. So we have hydrolyzable lipids are those that contain a functional group that will react with water. The functional group is usually an ester, and the list of compounds includes neutral fats, waxes, phospholipids, and glycolipids. This means that the functional groups are polar groups which can react with water. Then the non-hydrolyzable lipids are non-hydrolyzable lipids. They lack such functional groups which are polar. Example is vitamin includes the steroid hormones, progesterone, estrogen, testosterone, and the fat-soluble vitamins A, D, E, K, ADEK. Okay, so we have simple lipids that are esters, that fatty acids formed with various alcohols. Complex lipids are esters made up of fatty acids, alcohol, and other chemical compounds. Then precursor derived lipids. Let's pause here. Let's pause the first lecture here so that you can have a break.